Welcome back to our last session of the afternoon. We're going to be, in this last session, when my co-organizers and I were thinking a little bit about what we wanted to do with this last session, one of the things that we thought might be an interesting addition to the program would be to think a little bit about alternative methods for thinking about reproducibility and transparency, both within the context of social sciences, but also thinking about what we can learn from other disciplines. We have three panelists here today to talk about sort of these alternative approaches, and we're very interested to hear what they have to say. In addition, we're going to be returning to the format before, where they'll each give a short presentation, and we'll have some time at the end for questions and answers. And then afterwards, we'll have wine and cheese reception immediately following this. So please stick around. You'll have your chance to ask all of the panelists from today all the other questions you haven't had a chance to do. So I'd first like to introduce Jonathan Eisen. Dr. Eisen is a professor at the Genome Center at the University of California, Davis, and holds appointments in the Department of Evolution and Ecology, the College of Biological Sciences, and the Department of Medical Microbiology and Immunology at the School of Medicine. His research focuses on communities of microbes. His overarching research goal is to create a field guide to microbes, such as exists for birds and trees. He's widely known as the leading authority on the study of microbes based on DNA sequencing and the author of more than 300 papers on the topic. His research focuses on the mechanisms underlying the origin of novelty, how new processes and functions originate. Most of his work involves the use of high-throughput DNA sequencing methods to characterize microbes, and then the use of development of computational methods to analyze this type of data. In particular, his computational work is focused on phylogenomics, the integration of evolutionary analysis with genome analysis. Prior to moving to UC Davis, he was on the faculty of the Institute for Genomic Research in Rockville, Maryland. He earned his PhD in biological sciences from Stanford University and his AB in biology from Harvard College. And finally, he was elected to the American Academy of Microbiology in 2012. Please join me in welcoming Jonathan Eisen.
Ooh, I get to play with someone else's computer. I have a distinct aversion to slides, so forgive me if I'm going to look down at my notes here for a bit. So I thought with the introduction you may have sort of guessed, but I'm from the biology area, and I've been involved in the genomics field for about 15 to 20 years now, and the genomics field has been leading and not leading in some areas related to openness and transparency, and I'd be happy to talk about that in more detail in the discussion. That's not exactly what I'm going to focus on. I just want to go through like 5 sort of points about transparency and openness as it relates to some of the things that I've been doing, and biology in particular. And so I am somewhat of an open science evangelist. I have been a bit obsessed with open science activities in particular since about 2003. So for those of you who don't know, my brother was one of the co-founders of the Public Library of Science, and when they were working on starting the Public Library of Science, he kept badgering me to sign their petition, which is how the whole thing started, that you would, as a scientist, you wouldn't publish in or review in non-open access journals, and I didn't get the whole point. I sort of did, but I didn't. And my brother even sent the fax. Yeah, they were still using faxes then. The fax related to signing this petition to me to ask not if I would sign it, but if I could get Craig Venter to sign it, because Craig was the head of the institute where I worked and a very famous person in genomics. And then, I sort of just watched as this was happening, and I signed up. I was one of the founding editorial board members of their journal, PLOS Biology, and we published one of the first papers in that journal, and I, signed on the dotted line that openness was great, and didn't completely believe it until sort of 2 events. Did this go off? No. Until sort of 2 events. One was a family medical emergency where at 3 in the morning in a hospital across the street from where I worked, I needed to get access to the medical literature to try and make a decision about treatment that the doctors didn't know what to do, and I couldn't get access to the papers. And I lost it. I completely just couldn't believe that here was an actual case where someone was trained somewhat in science, where I could read the papers and maybe make sense out of them, and I couldn't get them. And I was logging in by modem on my computer from the hospital room, and connecting to the internet and trying to download papers and read them. And I suddenly realized what my brother had been sort of talking about for a while that I had never personally realized was important. And then the second event was I went to a meeting that was supposedly about the openness and transparency in the field of genomics. So there was something called the Bermuda Agreement that many of the big genome centers that were working on this public human genome project. So you may have seen like press conferences at the White House where people got up there and said, this is for the benefit of humanity, and we're releasing the data hourly for everybody to use, and it's this wonderful thing for everyone in the world. And there was a meeting to discuss the openness policies about genomic data called the Fort Lauderdale meeting that came up with this Fort Lauderdale Agreement, and I was the representative of our institute that went there. And at this meeting, I found out two things that were really interesting. One was that the human genome data was not, in fact, released openly. It was released, and you had to basically sign a caveat or agree to a caveat that said, you could look at this data and you can analyze one little piece of this data, but you can't analyze the whole data set until we publish our big genome paper in some unforeseen future. So at the White House, they said it's free and open. And here, the truth was that it wasn't completely open. And I was pretty stunned by this. So I went back after this meeting and after being convinced by some of the people there and I changed our institute's policies where we removed that line for many of our data sets and said, you can do anything you want with the data as soon as we release it. And this goes to some of the discussions that were happening before about the risks of openness. I was working on a very strange organism. It doesn't really matter what it is. A strange organism. We sequenced a large fraction of the genome of this organism using these DNA sequencing methods, and we posted the data and sent out an announcement to everybody who worked on this organism or its relatives. Here's the data. You can actually do anything you want with it. You don't need to wait until we publish a genome paper. And two things came from that. About 5 hours later, a friend of mine who worked on the organism sent an email saying you might want to remove the anthrax data from your data set. We were the place that sequenced the genome of anthrax for investigating the anthrax letters attacks, and some of the data from the wrong organism got put in our database. He told me he would never have looked at the data if we hadn't said that he could do it with no restrictions. And the second one was, we didn't get scooped on anything. We got like 75 people offering to help us on our paper about the genome of this organism. Without any pressure, without anything, they just said, look, I found this really cool thing. If you want to put it in your paper, go ahead. So I sort of became convinced of this, that the risks of openness, which I'll come back to right at the end, were not as big as we had thought. We had all feared that everybody was going to steal our data, and it turned out that instead people were actually collaborative and helpful and friendly. If you treated them with respect, as someone said this before, if you treated them with respect, usually it came back. And so I've been involved in many other sort of open movements. I've been an editor at many PLOS journals and at some new open access journals, including the Biology Archive, BioArchive, and there's lots of discussion in the biology community about requiring code be all open, available, open source code if you want to publish a paper, making it available before publication in some cases after. There's a big movement for complete openness of everything in many biological papers. That is sort of the full reproducibility issue. It's been not received incredibly positively by some aspects of the biology community and received pretty positively by other parts of the community. And it depends in part on the culture of the different field, I think, and it depends in part on the type of data that people are producing. So for example, there are some ecologists who have spent 30 years working on a field site and making observations, and when they write up a paper to have a journal editor tell them, you have to post every single recording you've ever made and put it in the paper, in the archive somehow, people are pretty resistant to doing that, especially since it's a relatively poorly funded field. And whereas in genomics, we've sort of accepted that if you get a big project to characterize the genome of an organism, you're going to be releasing the data before you even publish it, not only after you publish it. So there's sort of cultural differences within the field that probably mirror some of the cultural differences that you see in social sciences or between social sciences and other areas. So, and I'm happy to talk about this in the discussion about been involved in sort of open data and open software and open materials. So people are required to share strains of microbes if you publish a paper on that microbe, and that's getting pretty complicated. In the last few years, I've been a little bit more involved in sort of the transparency side of open science as opposed to just sharing things, but trying to make things more transparent. And I guess a confessional here. I was what was called the academic editor-in-chief of this journal, PLOS Biology, which was one of the sort of higher profile public library of science journals. And I mean, I like PLOS and I like PLOS Biology, but in the end, I actually quit over transparency. They had a policy where if people submitted a paper, there's a professional editors, like in Nature, Science, and Cell, that handle papers, and then they ask academic editors to help them edit and review those papers. If the paper is accepted, the academic editor is identified. If the paper is rejected, the academic editor is not identified. And I couldn't bear this. I just felt very non-transparent about the process. And so after doing that, I've sort of been sniffing around for ways to work on transparency. And one thing that I would like to propose, which I don't know how many people do in social sciences, is much more of a concern probably in biomedical sciences than it is in many other areas, but disclosures are a really important part of science and evaluating publications. And we handle it horribly, even in the biomedical area and certainly in any other area of biology research. And we really need to improve disclosures of potential conflicts of interest or real conflicts of interest. And I have sort of been fishing around for ways to do this in a way that is easily accessible. And I think there is a solution. I unfortunately missed the ORCID meeting the day before yesterday. They were here. So ORCID is this unique author identifier system. And I think if ORCID added a link entry where you could post your disclosures associated with your scholarly work, then anybody who sees a paper of yours, if your ORCID ID is there, they can rapidly track it to whatever your disclosures are. And so I've posted whatever I can think of for my disclosures there, and a bunch of other people have. But I think we need a system for more transparency about possible conflicts of interest among scholars. And then the last thing I want to just sort of briefly mention is this issue of risks and rewards. And despite being sort of a bit overly zealous of the benefits of openness and berating people about their lack of openness, I've come to realize that there, in fact, are real risks with openness, way more than I appreciated, and I think we need to deal with this. And I'll just give you 3 quick examples of this. Some of them are sort of trivial, some of them are a little more complicated. So in the biology field, in ecology in particular, there's been a growing appreciation that poachers are going to open access publications, getting GPS coordinates for endangered species and other organisms, and going and capturing them or killing them or advertising hunts for them. And so there's a movement to not include that information in scientific publications. And whether they're open or not publications, it doesn't matter. I mean, just to remove some information in the publication in a similar way to human subject research where you might remove identifiers. And so, this is one of those things where some journals have a law, basically, that says you must disclose everything. They're going to have to adapt their policy, or people will be basically not submitting papers to those journals anymore. So there's some sort of real risks that come associated with full reproducibility and full disclosure that's not just about human subject research. I'm sure this happens in archaeology and in a variety of other areas where people are going after artifacts. And in my field, there's a lot of movement where people go to a new site, collect an organism or an environmental sample, and then run DNA sequencing reactions on those samples, and you can get information about the genetic content of a rare plant or a land race for an agricultural crop or a new microbe from a particular place. And in the past, people posted this data without really feeling like it was a big risk, but now people can actually go to those data sets and synthesize parts of the organism in the laboratory, like proteins that they encode, and never need access to the organism. And there's a big biopiracy sort of movement associated with doing this, and there are a lot of countries and other institutes and agencies that are very worried now about publicly posting data from their sites where people who don't need access to the site or the organism can now start to capitalize in some way on that information. And I think there are many other examples of this, of like real risks associated with being open, and I'm starting to appreciate this a little more. And it goes to something someone said in the last session, which is, I think we really need to think about the rewards that come for doing things that we would like people to do more than we have been. There's been a lot of the stick going around where, you have to publish your data, or you have to make your software open source, or you have to, even make your papers open access. But I think to compensate or to deal with some of these risks, I think we need to think about the rewards. And some were mentioned, like, if you release your data, we need to make sure that data gets cited if it's used, right? I mean, so this does not actually happen. So in biology, there's big databases with all the DNA sequence data. If you use the data from those databases, very few people actually cite the original source of the data. And we could do a lot better in that area, and therefore ameliorate, I think, some of the risks that come from being more and more open, better tracking, altmetrics, whatever you want to think about. I think that there's a lot of need for coming up with better systems so that the junior scientists, in particular the junior scholars in whatever field, feel like they're not risking their careers by being as open as possible, and in fact that they benefit in some explicit way from doing it rather than in some possible theoretical way. And I think my opinion now is that rather than openness is all glory and shiny, which I still kind of think it is, but is that the default should be open. We should, rather than the default being sort of closed, the default should be open, and then in cases where there's a problem with being open, we need to figure out how to solve that to make things be more open. Rather than saying that openness is all perfect, we should accept that there are caveats and try and solve that. So I think that's all I wanted to say about this, and we can move on to the next person.